Hey guys, Miss Cote here, and this is video 4 of the biochemistry unit. Today we're going to finish off our chapter on macromolecules by discussing nucleic acids. So let's get started. In the previous video on proteins, you learned that the amino acid sequence that makes up a polypeptide is determined by a gene. Genes are made up of DNA, which is an example of a nucleic acid. The last class of biological molecules are the nucleic acids. There are two types of polymers of nucleic acids, deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. And yes, you should probably learn how to spell these words. DNA is stored in the nucleus of the cell and contains all of your genetic information, all the instructions to make all the proteins that makes you, you. And since it cannot leave the nucleus, RNA, specifically a type of RNA called messenger RNA, is able to bring the information out to the ribosomes in the cytoplasm to make the proteins. This whole process is called gene expression and will be covered in way more detail later on in the course. The monomer of a nucleic acid is a nucleotide. A nucleotide is made up of three parts: a nitrogenous base, a pentose sugar, and one or more phosphate groups. Before we get into the diagrams, here's another slide with some important vocabulary you're going to need to know. I'm not going to go through each one, so if you are trying to write this all down, I suggest you pause the video. If not, let's move on. So here's our nucleotide with the base, sugar, and phosphate group. Over on the left, it shows you how one nucleotide can be part of the bigger structure known as the polynucleotide. But first, let's look at the bases and sugar in a little more detail. The nitrogenous bases are split into two groups. The pyrimidines are one-ring structures, and that would be cytosine, thymine, and uracil. The purines are two fused ring structures, and those would be adenine and guanine. How I always remember it is that the bigger name has the smaller structure and vice versa. You don't need to memorize which base is a purine or pyrimidine, but it would be nice if you could recognize the structure and identify them as a nitrogenous base. Remember, only proteins and nucleic acids contain the element nitrogen, and this doesn't look like an amino acid. The pentose sugar is different between the two types of nucleic acids. DNA uses the sugar deoxyribose, while RNA uses ribose. So the nucleic acid is actually named for the sugar that it contains, and not that you need to memorize the structures. Note that the difference between the two sugars is at the second carbon. So in joining the base to the sugar, we have a nucleoside. All that's left to do is add the phosphate group, which is added to the 5 carbon of the sugar, and there you have your nucleotide or nucleoside monophosphate. Like the other biological molecules, joining the monomers together involves dehydration reactions. A covalent bond, called a phosphodiester linkage, another type of bond you need to know in AP, is formed between the sugar and the phosphate group. The phosphate is added to the hydroxyl group on the 3' carbon, which gives the chain 5' and 3' ends. 5' and 3' are not that important now, but you will need to know this later on. This results in a repeating pattern of sugar phosphate units that's referred to as the sugar phosphate backbone. Those yellow nitrogenous bases stick out to the side. I'm sure you're familiar with the overall structures of DNA and RNA, which is something you needed to learn for MCAS, but just in case, let's do a quick review. DNA is made up of 2 polynucleotide strands that twist around each other, forming a double helix. The 2 strands run anti-parallel to each other, where one strand runs in a 5' to 3' direction, while the other runs in a 3' to 5' direction. Again, more on the importance of that when we get into more detail on DNA. The two strands are held together by hydrogen bonding between the bases. Note the 3 dots. And remember that there's a method to the base pairing. Adenine pairs with thymine, and cytosine pairs with guanine. This is called complementary base pairing. As we will see later on, this makes it very easy for DNA to make copies of itself. We've already discussed that one of the differences between DNA and RNA is the sugar in the nucleotide. RNA uses ribose. A second difference is that RNA is single-stranded as opposed to the 2 strands that make up DNA. However, if the RNA molecule is long enough, complementary base pairing can happen within the same molecule. This can lead to an RNA molecule that takes on various shapes, such as transfer RNA. Another major difference is that the base thymine is not found in RNA. Instead, another base called uracil takes its place and will complementary base pair with adenine. What's interesting to note that in regular bio you learned about the 3 types of RNA: mRNA, tRNA, and rRNA. Just know that there are other types of RNA that have some pretty cool functions, and we'll learn more about those in the gene expression unit. So that's it for the nucleic acids in Chapter 5 material. The next and last episode for this unit is going to be on enzymes. If you have any questions, please feel free to bring them up in class. So until next time, let's be good humans.